Good afternoon, students. We are discussing about non-Mendelian inheritance, and in our last class we had discussed about incomplete dominance. So today we'll start with codominance. Okay? So codominance. In certain cases, both the dominant and recessive alleles lack their dominant and recessive relationship, and both of them have the capability to express phenotypically in the heterozygous condition. But in heterozygous condition, the dominant and recessive alleles remain side by side. The heterozygous offspring of F1 hybrid produce F2 offsprings in the phenotypic and genotypic ratio of 1:2:1 as found in incomplete dominance. So, what is codominance? When the two alleles neither so dominant nor the recessive relationship, or they never show intermediate condition, but both of them express themselves simultaneously, then it is known as codominance. Okay? যেতিয়া দুটা এলিলছৰ কোনোটোৱে নিজৰ কেৰেক্টাৰ ইণ্ডিভিজুৱেলি এক্সপ্ৰেছ নকৰে, প্ৰকাশ নকৰে, নাইডাৰ ডমিনেণ্ট নৰ ৰিচেছিভ, বা সিহঁতৰ মাজত এটা মিক্স হৈ পেলাই কেৰেক্টাৰ এক্সপ্ৰেছ নহয়, কিন্তু দুইটা কেৰেক্টাৰে ছাইড বাই ছাইড এক্সপ্ৰেছ হয়, তেতিয়া সেইটো কোডমিনেন্স বুলি কয়। চ' কোডমিনেন্সত কি হয়, দুইটা কেৰেক্টাৰে এটা এলিল পেয়াৰত থকা দুইটা জিনৰ কেৰেক্টাৰে ছাইড বাই ছাইড এক্সপ্ৰেছ হয়। Both alleles which when present together in heterozygous individual express their traits independently instead of showing dominant recessive relationship are called codominant alleles. Next is multiple alleles. So the multiple alleles can be defined as a set of 3, 4 or more alternative forms of a gene which have arisen as a result of mutation of the normal gene and which occupy the same locus in the homologous chromosome. Okay, so multiple alleles means these are the alternative forms of a gene. It may be 3, 4 or more. Okay, generally what happen in alleles, one pair of genes are present, isn't it? Eta allele pairot duta gene thake, allele buli kole tate duta gene ekelege thake, kintu multiple allelot ki hoy, more than 2 alternative forms of a gene are present. Gene number 3টা 4টা 5টা বা তাতকে বেছি হ'ব পাৰে। So multiple allele, the best example of multiple allelism is again can be explained with the help of human ABO blood grouping, because in ABO blood grouping there are 4 alternative forms of the gene I, as I have explained in codominance. So codominance already explained korisu je I gene 2, which is responsible for human blood grouping, it has 3 different alleles, 3টা বেলেগ বেলেগ allele থাকে, IA, IB and I naught. So this is again the example of multiple allelism. Okay, now we'll discuss law of independent assortment. It is the inheritance of two genes. It is also known as Mendel's third law. Okay, so Mendel's first two laws in law of dominance and law of segregation, these two were based on Mendel's monohybridization experiment, but this law of independent assortment, it is based on Mendel's dihybridization experiment. So what is law of independent assortment? Mendel's law of independent assortment is the outcome of dihybrid cross. According to this law, if the inheritance of more than one pair of characters is considered in a hybridization experiment simultaneously, the factors or genes for each pair of characters assort independently of the other pairs. In his experiment, he made a cross between pea plants having yellow cotyledon color and round seed surface and the plants having green cotyledon color and rough or wrinkled seed surface. All the F1 hybrids of this cross were phenotypically with yellow cotyledon and round seed surface. So I am showing how. Okay. So here I'll show the complete chart. So these are the phenotypes and we will get 9 round and yellow plant, yellow wrinkled tree, green round tree plants and green wrinkled one. So total will be 16 as we have made 16 boxes here. Okay, so this is how dihybrid cross is performed. Okay, so this will give the phenotypic ratio nine is to 3 is to 3 is to 1. Okay, so phenotypic ratio to over 9 is to 3 is to 3 is to 1. And again the genotypic ratio of this cross will be 1 is to 2 is to 2 is to 4, 1, 2, 1, 2, 1. So total will be 16 because that is genotypic ratio to keneke 1 is to 2 is to 2 is to 4 is to 1 to 1 to 1 hoise because suppose yellow and round. So for yellow round combination, the different genotypes are there. Yellow round, capital Y, capital Y, capital R, capital R. Then for yellow round, capital Y, capital Y, capital R, small r. Again for yellow round, small y, capital R, capital R. Then capital Y, capital R, small r. So beleg combination of phenotype eke hoy. So heneke kimanta capital Y, capital R ase, kimanta capital Y, capital R, small r ase, hetu basi uliai ami ei genotypic ratio to pam. Okay, so this is the law of independent assortment which can be explained with the help of dihybrid cross. Okay. So I'm repeating again, according to the law of segregation, the factors for pairs of characters not only segregate but assort independently and may combine randomly with either of another pair during the formation of gametes. So independent assortmentot যি কম্বিনেশ্যনে নাথাকক সিহঁত ইণ্ডিপেণ্ডেণ্ট হয় আৰু গেমেট ফৰ্মেশ্যনৰ টাইমত সিহঁতে ইণ্ডিপেণ্ডেণ্টলি ইটোৰ লগত সিটো কম্বাইন কৰিব পাৰে। চ' if we see that law of segregation and law of independent assortment, actually both are same, but the difference is law of segregation is based on Mendel's monohybridization experiment, but law of independent assortment or Mendel's third law, it is based on Mendel's dihybridization experiment. So this is the difference, otherwise both the laws are same. Okay, so students, just go through it, practice it, means try to make the table of dihybrid cross from your own, then you will be able to understand and again find the genotypic and phenotypic ratio. Okay, so go through it. If there is any confusion, then we'll again discuss. Thank you.